Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from Department of Biochemistry, Independent Medical College, Faisalabad. This is my third lecture on transcription. In my previous two lectures, I discussed about the RNA synthesis in prokaryotes and eukaryotes. Now in this lecture, we will study about the post-transcriptional modification of RNA in prokaryotes as well as in eukaryotes. Primary transcript is the initial linear RNA copy of transcription unit, and this transcript is only the segment of the DNA between the initial and termination sequence. This transcript will undergo post-transcriptional modification. In ribosomal RNA and transfer RNA, it is cleaved by, this primary transcript is cleaved by the ribonucleases, and for the transfer RNA it is further modified. We will study in the next slides that how transfer RNA, mRNA and ribosomal RNA, they will undergo transcriptional modification, post-transcriptional modification. Prokaryotic mRNA is almost identical to its primary transcript, but the eukaryotic mRNA is entirely different. It is extensively modified. First of all, how ribosomal RNA it is modified. We have studied that the 28S, 18S and 5.8S ribosomal RNA, they are synthesized in the nucleolus and they are transcribed by the RNA polymerase 1. But the 5S ribosomal RNA, they are not synthesized by RNA polymerase 1, rather it is transcribed by RNA polymerase 3. So here you are seeing that 5S ribosomal RNA is synthesized separately, and other species of the ribosomal RNA, it is synthesized separately. They are present in the they are synthesized in the nucleolus, and they are assembled with the proteins within the nucleolus, and then there is the further modification of the bases as well as the ribose sugar. Here it is written that methylation of bases occurs. This then there is the further trimming of the ribosomal RNA into the 5S RNA, into the 41S RNA. It is the 18S ribosomal RNA. It is processed separately in the in the proteins, and the other species of the RNA, they are processed separately. These species of the ribosomal RNA are the 5.8S, 28S and the 5S ribosomal RNA. When they are trimmed into the required size, they are exported out from the nuclear pore into the cytoplasm, and the smaller S, which is in the eukaryote, this is the 40S ribosomal subunit, and this is the 60S ribosomal subunit. It assembled into the functional 80S ribosomes. This is again the same presentation, that how the polymerase 1 synthesizes the pre-ribosomal RNA, which from the same transcript, 28S, 5.8S and 18S ribosomal RNA, they are synthesized, and which are then cleaved by the RNases to form the 28S, 5.8S and 18S ribosomal RNA. This modification within the nucleus, within the nucleolar region, it is facilitated by the SnRNA, and after their formation their modification, their trimming, the proteins become the component of the ribosome associated with the pre-ribosomal RNA prior to or and during its modification. Now the transfer RNA. Transfer RNA, it is both eukaryotes, in the both eukaryotic and prokaryotic transfer RNA, they are made from the longer precursor molecules, and they are extensively modified. First of all, see that in the 5' end, 16 nucleotide sequence at the 5' end, which is you are seeing that they are shown in the green color. They are cleaved by RNase P. RNase P, it is a type of ribozyme RNA which act as enzyme. So this 16 nucleotide, it is removed. Another purple color or blue color part, it is the intronic region. This 14 nucleotide intron is removed by nucleases. Then at the 3' end, CCA sequence is added by removal of the uracil residues at the 3' end. And another change in the transfer RNA is that the many bases, they are converted to the characteristic modified bases. These are shown in the yellow colored boxes. These nucleotides which are changed or which are modified bases, they may be the ribothamidine, pseudouridine and dihydrouridine, present in the variable loop, present in the this TYC loop, anticodon loop, and this is the D loop. This is the same picture for the transfer RNA, that the longer precursor of the transfer RNA, it is synthesized. Intronic region, the blue color is the intronic region, it is removed, and the few regions in the 5' and 3' end, they are removed, and there is the modification of the bases, and also there is the addition of CCA, and after all its modification, it is exported out, and the mature transfer RNA is now present in the cytoplasm. Now the mRNA. mRNA is synthesized in the nucleus by the RNA polymerase 2, and when it is synthesized, it is called the heterogeneous nuclear RNA. Heterogeneous nuclear RNA. This. And what are the modifications which occurs? This is the addition of the 5' cap, addition of the poly-A tail, and removal of the introns. They may be modifications may during these modifications can be the co and post transcriptional modification in the nucleus, and then there is the formation of mature mRNA, which is ready to be translated into the proteins. So we will see that in detail that how mRNA it is in the eukaryotes, how mRNA it is modified. First of all, the 5' capping. 5' capping require removal of gamma phosphate from the 5' triphosphate of the pre-mRNA. Okay, the terminal phosphate group is removed. Terminal phosphate group is removed, and then there is the addition of GMP from GTP. The second step, methylation of guanine, which will often occurs in cytosol, and this is catalyzed by the guanine 7-methyltransferase. And S-adenosyl methionine is the source of methyl group. Why this 5' cap has been added, or what are the functions for the addition of 5' capping? It regulate the nuclear export. It prevents the degradation by the exonucleases, promotion of translation, and promotion of 5' proximal intron exons. So these are the functions which are provided by the 5' cap. This is a very excellent picture for the how the 5' cap it has been added. The upper purple color is the 5' cap with and this cap is the cap portion and this is the RNA portion. First of all, see that there was the 3 phosphate groups at the right hand side, and at the top of the picture see that the 2 phosphates alpha and beta, 2 phosphates, they are of green color, and gamma phosphate, red color phosphate, this is the gamma phosphate. It has been removed by the phosphohydrolase. And GTP, this is the GTP is added, and pyrophosphate is removed. Guanylyl transferase is the enzyme, and this structure is formed. Now this phosphate, the yellow color phosphate, is from the GTP. Then there is the methylation of the guanine, and this is done by the guanylyl 7-methyl transferase. S-adenosyl methionine is the source for the methyl group, and so this is the mature cap which has been added. 7-methyl triphosphate cap, it has been added. This is the cap region, and this is the mRNA. And also this linkage, the formation of cap linkage is 5' to 5' linkage. Now how the poly-A tail is added? Poly-A tail is added, see that this is the polyadenylation signal sequence. This is the polyadenylation signal sequence. After the polyadenylation signal sequence, there is the cleavage site, and then there is the string of U's. At the cleavage site, the mRNA, it is cleaved, and the these adenine, they are added by the poly-A polymerase. ATP is the source for the phosphate group, and the repeat of ATP addition, and there is the pyrophosphate is the released. About 250, 40 to 250 introns, 250 adenine nucleotides, they are added beyond the signal sequence. And signal sequence, I have told you before that this is the AAUAA. This is the signal sequence. Now the third change which occur in the mRNA is the removal of introns. The introns, they are the intervening sequences which are not the coding sequences. They are removed by the way of splicing. And the complex which accomplishes this task is the spliceosome. Spliceosome is the complex which contains 5 SnRNPs. 5 SnRNPs, small nuclear ribonucleoproteins. They are the U1, U2, U4, U5 and U6. They are uracil rich SnRNA, uracil rich molecules. And there are other about 60 proteins which are involved, and in the spliceosomes there is the RRM. RRM means RNA recognition motif, and also the serine arginine motif. So all these things, they make up the spliceosomes. These 5 RNAs, RRM and SR containing proteins form a small nuclear ribonucleoprotein, and this is called the SnRNP complex. I have told you before that SnRNAs, they are uracil rich small nuclear RNA. They are also called the SNRNAs, and they base pair with the consensus sequences and thus help in the removal of introns. How they help in the removal of introns? The first of all, these SnRNPs, they bind with the sequence where the introns they have they are to be removed. The 2' OH group of the adenine, here see that the yellow color portion, this is the intron. A will be involved in the formation of gradient structure A, and the blue color axon 1, and the next is the axon 2. These blue color axons, they will be joined at the end, and this yellow color portion, this is the intronic region, and it will be removed. The green color oval like balls, they are the SnRNPs. The primary transcript combine with the SnRNPs to form the spliceosomes. This is the spliceosomes. Here what happens, that the 2' OH of the branch site, OH of the branch site attack the 5' phosphate at the splice donor site of the introns, forming the 2 to 5' phosphodiester bond. I'm talking about this 2' OH of the branch site. This one attacks this 5' phosphate at the splice donor site, again in the introns, forming the 2 to 5' phosphodiester bond. 2 to 5' phosphodiester bond. Okay, now this 3' OH group is free, and it will attack at the 5' phosphate at the splice acceptor site, forming a phosphodiester bond, and by this joining, the axon 1 and axon 2, they will be joined. SnRNPs, they are removed for the further use, and the excised introns in the form of gradient structure, it is freed. This is another diagram which shows the same mechanism, that how these uracil rich molecules they are involved in formation of the mature mRNA containing only the axons, and the intronic regions, they are removed. You can use either of the diagram which you find it easy, and this is the again the sequence which shows that this GU the regions which are present at the 3' and the 5' end of the axons and the introns. This GU and AG regions, they are invariant structures, and these are the consensus sequence which are almost always present at these specific sites. This is the intronic region which is removed, and these are the axons which will be joined. SLE, systemic lupus erythematosus. This is an autoimmune disease characterized by a particular spectrum of autoantibodies. Autoantibodies, they are produced in SLE, and they are produced against many of the cellular components, including the chromatin, ribonucleoprotein, and cell membrane phospholipid. But most important for in all of 3 components against which the antibodies are produced, they are the SnRNPs. And in fact, this is the same SnRNPs which were discovered because of the study using the antibodies obtained from the patients with SLE. So most importantly, the antibodies, they are produced against these SnRNPs. This is the most common symptoms for the SLE patient, and in which the most common feature, this is the and distinguishing feature, this is the butterfly rash present on the face of the patient. And there are also other psychological and systemic symptoms, which include the fever, photosensitivity, ulcers in the mouth and nose, muscle aches, and there is the inflammation in pleura and pericardium, and there is the poor circulation around the body. Splice site mutation. Over 20% of all the genetic disease, they are the result of mutations that affect RNA splicing. This splicing, the splice site mutation, can be either due to the exon being skipped or the introns being retained. Exons being skipped or the introns, they are not removed. And example is the beta thalassemia. Beta thalassemia is the disease which occurs due to the mutations in the intron exon junction. There is a change in the nucleotide at the intron exon junction, and due to this the beta chain is underexpressed. This is not produced, beta chain is not produced, but the alpha chains, they are produced at normal rate, but their tetrameric structure, it is disturbed. That's why this hemoglobin is not properly formed, and it is severely underexpressed. They can also activate cryptic splice site, which are the sites that contain the 5 or 3 consensus sequences but are not normally used. Now, how the diverse set of proteins, they are formed from single RNA. So these are the same RNA, right? And here you are seeing that 2 types of the mRNA, mature mRNA, which has been produced due to the alternative splicing. And example, you can see that in this RNA, all the exons, they are joined, and in the second form of the RNA, only 2 exons, they are joined. So this is the production of diverse set of proteins from limited set of genes. And example is the mRNA for tropomyosin. Tropomyosin is an active filament binding protein of the cytoskeleton and of the contractile apparatus in the muscle cells. This mRNA, the mRNA of tropomyosin, undergo extensive tissue-specific alternative splicing. So according to the tissues, they are spliced, and with the production of multiple isoforms of tropomyosin. This is the diagram again, which shows the mechanism of alternative processing of mRNA precursor. This form of mRNA processing involves either the selective inclusion or exclusion of introns, the use of alternative 5' donor site or alternative 3' acceptor site, and the use of different polyadenylation sites. So they are the mechanism for alternative processing of mRNA from the same mRNA precursors for different diverse set of proteins, diverse set of mRNAs, they are produced. Now there is the turn of the processing of microRNA and silencing RNA. MicroRNA and silencing RNA. The microRNA, first of all, it is produced by it is transcribed by the RNA polymerase 2 with the cap at the 5' end and the adenines, the poly A tail at the 3' end, and this occurs within the nucleus. And after the processing and with the help of proteins, which is the DGCR8 nuclease, and the processing, there is the trimming of the molecule. Before, it was the primary microRNA. Before the processing, which was the RNA, the microRNA, which was produced by the RNA polymerase II, it was the primary microRNA or pri-miRNA. After the processing and by the action of DROSHA DGCR8 nucleases, which trims the sequence both from the 5' and 3' ends, and here now it is pre-miRNA is generated. The partially processed, now this is the partially processed double-stranded RNA. It is exported out into the cytoplasm by the nuclear pore and by the help of exportin 5. This pre-miRNA is further trimmed by the action of heterodimeric nuclease dicer protein, and it will be trimmed by the dicer protein. And now there is the formation of 21-22 nucleotide microRNA duplex. This is one of the two strands. Here you are seeing that one is the blue color strand in the microRNA and in the duplex, and other is the green color. One of the two strand, here the green color strand, it will generate the mature RISC complex, forming the functional mRNA. This mRNA is loaded on the RISC complex, and this RISC complex containing the mRNA and which also contains the agoproteins, they are loaded on the mRNA, messenger RNA. And what it will do, it can degrade mRNA by one of the three ways. Either it will repress the translational machinery, the number second, it will do the de-adenylation. Adenine molecules, adenine residues, they are removed from the poly A tail. In this way also the mRNA will be deactivated and it will be not able to produce the mRNA proteins. And the third mechanism by which microRNA will inhibit or it will degrade mRNA is by the way that it will break down the mRNA into small pieces, and this way also the mRNA will be degraded and it will be not it is not able to translate it into the proteins. So this is the same explanation which I have told you in the previous diagram. This is the formation of silencing RNA. Silencing RNA pathway generates functional siRNA from large double-stranded RNA. They may be formed intracellularly by the RNA hybridization or by the extracellular sources, for example the attack of RNA viruses. These double-stranded RNA again undergo process, and about 22 nucleotide siRNA, which is again the double-stranded RNA segments, and by the way of dicer nucleases, they are loaded onto the AGO2 containing RISC complex, RNA induced silencing complex. RISC complex mean RNA induced silencing complex, and again one of the two strand in this complex in the about 22 nucleotide duplex, one of the two strands, it is selected to generate silencing RNA, and it will locate the target RNA, and it will do the silencing of the mRNA degradation by inactivating it. Now this is the function of reverse transcriptase enzyme, which is the reverse transcriptase enzyme which is used by the retroviruses. I have discussed this in the my previous lectures of the DNA. This is the diagram which explain that how the HIV they are attack on the cell membrane, and what are the receptors, chemoreceptors, CD4 receptors, they are used by the viruses, and how these viral DNA it is integrated into the mammalian RNA, mammalian DNA, and when it is integrated into the DNA, it will there will be the transcription and the production of 3 types of the proteins. They are Rev, Nef and Tat. Now what are the functions of Rev, Nef and Tat? This is your assignment. You will explain this diagram and submit me the assignment which specifically shows the function of reverse transcriptase, how AIDS retroviruses or the viruses of AIDS they produces its effects, and what are the functions of the proteins which I have told you before, the Rev, Nef and Tat. You can also consult my these diagrams and the slides. You can also mention these facts also. The second part of your assignment is this is the MCQs. You will give the correct answer with the logic. And these are some short questions. How you will submit your assignment is that you will prepare your both chapters, DNA 1 and the RNA 1, the both chapters of the DNA and RNA, and this will be you will now close your books and read my assignment, read my questions, and attempt these questions as you are sitting in the examination hall. Do not copy it from either of the notes or books or someone other's assignment. You will submit your assignment within the 10 days of the uploading of this lecture. Now your 2 of your chapters in the genetics, RNA and the DNA synthesis, it has been completed. Best wishes and good luck.